Bismillahir Rahmanir Rahim. I am Abu Zar Ghaffari Dar, student of MS Chemistry, University of Management and Technology, Sialkot campus. I'm highly grateful to our teacher, Ma'am Tabinda, for providing me an opportunity to present the topic Fourier transform infrared spectroscopy. Fourier transform infrared (FTIR) spectroscopy is a powerful analytical technique used to identify and quantify different molecular components in a sample. FTIR spectroscopy is widely used in many fields such as chemistry, material science, and biology. Now we will discuss the principles of FTIR spectroscopy. It is based on the interaction of infrared radiation with molecules. When infrared radiation is passed through a sample, certain frequencies are absorbed by the molecule, causing them to vibrate. These vibrations are specific to the functional groups present in the molecule, which can be used for identification. Molecules can vibrate in different modes such as stretching, bending, and twisting, each of which corresponds to a specific frequency of infrared radiation. When infrared radiation is passed through a sample, the resulting absorption spectrum provides information about the functional groups and structure of the molecule. The Fourier transform is a mathematical technique used to convert the interferogram obtained from the FTIR spectrometer into an absorption spectrum, which is a plot of absorbance versus frequency. Now we will discuss instrumentation and components of an FTIR spectrometer. An FTIR spectrometer consists of several key components that work together to produce an infrared spectrum. The major components include source, interferometer, sample compartment, detector, and computer. The source emits a broad spectrum of IR radiation that is passed through the sample. Typical sources include a globe, silicon carbide, or a ceramic source. The interferometer splits the infrared beam into two paths and then recombines them, creating an interference pattern known as interferogram. Michelson interferometer is the most common type of interferometer used in FTIR spectroscopy. The detector measures the intensity of the IR radiation that passes through the sample. Common detectors include DTGS, deuterated triglycine sulfate, and MCT, mercury cadmium telluride. Now we'll discuss sample preparation techniques for FTIR analysis. The choice of sample preparation technique depends on the nature of the sample and the specific information that needs to be obtained. For solid samples, they can be prepared as pellets, films, or powder using different techniques. Liquid samples can be analyzed using a liquid cell or by placing a drop of a salt plate. For gaseous sample, they can be analyzed using a gas cell where the gas is enclosed in a sealed chamber with windows that allow the infrared radiation to pass through. Qualitative analysis using FTIR spectroscopy. As we all know that FTIR spectroscopy is a powerful tool for identifying unknown substances, as each molecule has a unique infrared absorption spectrum that acts as a fingerprint. Comparing the spectrum of an unknown sample to a database of known spectra can identify the substance. FTIR spectroscopy can be used to identify functional groups present in a molecule, such as carbonyl, hydroxyl, and amino group, providing information about the molecule's structure. FTIR spectroscopy can be used to analyze complex mixture and identify the different components present. In quantitative analysis, FTIR spectroscopy can be used to measure the concentration of specific components in a sample by analyzing the intensities of the absorption peaks. Beer-Lambert law. The relationship between the absorbance and concentration is governed by this law. Calibration standards with known concentrations are used to develop a calibration curve relating absorbance to concentration. Once the calibration curve is established, the concentration of an unknown sample can be determined by using its absorbance and using the calibration curve. So FTIR spectroscopy is used in quantitative analysis. Now we will discuss applications of FTIR spectroscopy in various fields. FTIR spectroscopy has a wide range of application in various fields including chemistry, material science, biology and medicine. In chemistry, it is used to identify and characterize organic and inorganic compounds. In material science, it is widely used in analyzing the composition and structure of polymers, ceramics and composites. In biology, it is used for studying the structure and function of biological molecules such as proteins and carbohydrates. Now we'll discuss its advantages and limitation. FTIR spectroscopy offers numerous advantages, making it a valuable analytical technique. However, it also has some limitations. Some of its advantages include versatility, sensitivity, speed, non-destructive nature, wide range of applications. And its limitations are that FTIR spectroscopy has limited sensitivity for some compounds, interference from water, limited information about specific atomic positions, and it can be complex to interpret. Now, data acquisition and processing in an FTIR spectroscopy. The data acquired from an FTIR spectrometer is in the form of an interferogram, which needs to be processed to obtain an absorption spectrum. Interferogram acquisition is a time domain signal that represents the interference pattern of the infrared radiation. Fourier transform is a mathematical operation that converts the interferogram from the time domain to the frequency domain. The resulting absorption spectrum is a plot of absorbance versus frequency, which provides information about the functional groups and structure of the molecule. Now, at the end, our last presentation is recent developments and future trends in FTIR spectroscopy. FTIR spectroscopy continues to evolve with advancement in instrumentation, software, and applications. Future trends include the development of miniaturized and portable FTIR spectrometers, coupled with other techniques such as mass spectrometry and the use of machine learning for data analysis and interpretation. Portable FTIR spectrometers. Miniaturized and portable FTIR spectrometers are being developed for use in various fields, including environmental monitoring, food safety, and medical diagnostics. FTIR MS coupling. Coupling FTIR spectroscopy with mass spectrometry provides complementary information about the molecular structure and composition of samples, enhancing analytical capabilities. Machine learning in FTIR. Machine learning algorithms are being employed for data analysis and interpretation, enabling faster and more accurate identification and quantification of components in complex mixture. At the end of this presentation, I would like to thank you all for having a patience hearing. Thank you very much.